Welcome to the Attention Deficit Disorder Expert Podcast Series by Attitude Magazine.
Hello, everyone. I'm Carol Fleck, and on behalf of the Attitude team, I'm pleased to welcome you to today's ADHD Experts presentation titled Epigenetics, Understanding Its Role in ADHD and Future Applications. Leading today's presentation is Dr. Charlotte Cecil. Dr. Cecil works at the Erasmus Medical Center located in the Netherlands. She is an associate professor in biological psychopathology at the Department of Child and Adolescent Psychiatry and head of the In-Depth Lab. Her research focuses on the environmental and genetic factors that come together to influence a baby's brain development and mental health from the time they're born through adolescence. By identifying these factors early on, clinicians have a better chance to possibly detect, prevent, or treat chronic mental illness later in life. In today's webinar, we'll discuss the complicated and fascinating relationship between genes and the environment. This relationship is called epigenetics, and through this research, we now know that a person's exposure to pollution, chronic stress, trauma, smoking, and other factors can influence a baby's development even before they're born. Today, we'll learn more about epigenetics and what it can tell us about our future. We'd like to begin today's webinar by asking this poll question to our live audience. What do you hope epigenetic research will one day lead to? Please select your answers and comment in the text box under the video player to tell us more. For answers to common webinar questions about slides, transcripts, and certificates of attendance, click on the FAQ tab on your webinar screen. If you're listening in replay or podcast mode, visit attitudemag.com and search podcast 473 to access the webinar resources, or simply click on the episode description wherever you stream your podcasts. And if you support the work we're doing here at Attitude to strengthen the ADHD community, we encourage you to visit attitudemag.com/subscribe and sign up for Attitude Magazine. Our latest Success at School issue has lots of great advice for parents whose children learn differently. And for adults looking to impress their managers and kill it at work, we've got great advice for you too. So sign up today for Attitude Magazine for yourself or to share with a teacher or a loved one who could benefit from greater ADHD understanding. Click the magazine tab on screen to learn more. So without further ado, I'm so pleased to welcome Dr. Charlotte Cecil. Thank you so much for joining us today and for leading this discussion.
Thank you so much, Carol, for having me here today. I'm really excited to be able to share some of the latest findings on epigenetics and ADHD with the Attitude community. And I'm curious to hear about your thoughts and questions as well. So before I get started and tell you a bit more about epigenetics, I thought that it would be useful to take a step back and give you a bit of context in terms of reflecting on what do we currently know about the causes of ADHD and what do we not know yet. So I think by now there's strong scientific consensus that much like most complex human characteristics and behaviors and traits, ADHD is likely to reflect really the influence of both a person's genetics as well as their environment, beginning as early as in the in utero environment. So, for example, genetic studies have shown that ADHD tends to run in families, so it tends to have quite a moderate to high degree of heritability based on things such as looking at twin studies or family studies. And studies that have tried to pinpoint the actual genes that may be responsible have shown that ADHD is really polygenic. So what we mean is that in the vast majority of cases, this is not the result of a single gene, but really a combination of many different genes, some variants of which are more common in the population, some of which are rarer, and together they can increase or decrease the likelihood of developing ADHD. And this actually, the combination of these genes might differ from person to person. So it's already very complicated when we just look at genetics, but in addition, A lot of studies so far, for example, epidemiological studies have shown that the environment also likely plays an important role. And while there's been a lot of different factors associated with an increased risk for ADHD, I think for now the strongest evidence is really centered around the prenatal environment and early neonatal factors. I want to just stress here that a lot of these factors, it's unclear to what extent they're really causal. We are making advances in that respect and trying to single out what are really causal factors. But in terms of associations, I think some of the most robust factors are things such as maternal cardiometabolic conditions in pregnancy, stress exposure, inflammation, smoking, and also exposure to chemicals such as lead, as well as birth-related factors such as being a lower birth weight or more premature, so a lower gestational age. So there's been a lot of research trying to map what factors are involved, but the reality is that no single factor, either genetic or environmental, at the moment has really emerged to be either necessary or sufficient to explain why a given individual might develop ADHD, and particularly any impairment related to that. One of the complications is that actually a lot of these factors that have been identified, they're not that specific to ADHD. Actually, because they might involve, act on brain development, they tend to be related to also other types of outcomes, such as cognitive or neuropsychiatric outcomes, not only ADHD. And because of that, because they tend to have quite small effects and be not very specific to ADHD, at the moment, they're not very useful clinically in terms of really predicting early on, before the onset of ADHD, who might ultimately develop ADHD. And I think one of the biggest stumbling blocks at the moment in the research, or one of the key barriers, is the fact that even though we know genes and the environment are important, actually the vast majority of research still looks at either genes or the environment. So you really have your genetic studies on ADHD and you have your environmental studies on ADHD. But what we know that actually it's very difficult to separate these from one another, and they're constantly working together. So just to give you a concrete example, imagine you have two identical twins. Now these are genetically almost identical. And imagine that they have the same genetic risk for, say, cardiovascular disease. But you can imagine that if one of these twins is eating very poorly, not exercising, smoking, etc., they will be much more likely for that genetic risk to actually manifest compared to the other twin who has the same genetic risk, but it is leading a healthy lifestyle and exercising and all of that. So this genetic risk is not the full picture. You usually need a certain environment for that risk to actually manifest or not. And the same kind of goes for the environment. So you can imagine that, if you put 10 people in a room and you expose them to exactly the same environmental condition, they might probably react quite differently from one another. And that's likely because they're also genetically quite different from one another. So your genetics can also make you more or less responsive to a given environment. So we really need to be looking at these factors together. And how they work together at different points of development to shape a child's health and potential risk for something like ADHD, for example. And I think one of the limitations here has been that we just haven't been able to pinpoint very clearly how it is possible biologically that your genes and your environments come together. What is the mechanism? And this is really where epigenetics comes into the picture, I think, because it is emerging as one of those key mechanisms that through which the genes and environments are actually coming together to influence health and development. So I just wanted to, before I tell you how it might relate or not to ADHD, just tell you what it is in the first place for those of you who might not be familiar. So epigenetics is actually not one thing. It's a collection of things. It's an umbrella term that refers to different types of mechanisms. And you can think of these as sort of different types of chemical tags. And these chemical tags, they regulate where in your body and when in time or in your lifetime, your genes are expressed. So by expressed, I mean whether they're switched on or off, or you can think of it usually as more of a volume tuner, to what extent is that gene really being activated or deactivated. And these tags, they do this not by actually changing your DNA sequence at all, but by sticking to the DNA sequence. So to show you what I mean, so if we take the example of where in the body genes are expressed, I don't know if you can see my cursor, but basically, you can think of yourself and you know that you are a collection of different tissues and organs. And we also know that these tissues, they contain different types of cells. And you can see here, for example, that a skin cell is very different from a muscle cell and very different from, say, a brain cell. They have different shapes. They perform different functions. They're just really, really different. However, all of these cells have access to exactly your same DNA sequence. You have one DNA sequence, you're born with it, it shouldn't change throughout your life, and every cell has access to it. So how is it possible that these cells are able to read from the same set of instructions exactly what they need to become the type of cell that they are and perform the function that they need? Well, that's because these chemical tags, and I think the best recognized and studied at the moment, it's this tag called methyl. So it's DNA methylation is the process where these methyl molecules are sticking to the DNA itself. They're not changing the sequence, but depending on how many of these sticky things there are and where they are, it's like a signpost. It's telling cells which bits of your DNA should be read, so which genes should be switched on and which genes should be switched off. So that will be very different in a skin cell. The type of signposts that are there will be in different locations than, say, in a brain cell. And this is how your body is able to use this set of genetic information more flexibly in different parts of your body. But I also mentioned regulating when in time genes are expressed, because same goes for a person's lifetime. The DNA sequence itself does not change or shouldn't change, except if you have mutations and things like that. That's not good. Generally, your DNA sequence should not change throughout your lifetime, but the way that is used, the way that these genes are switched on and off can be very different. And this is actually what allows you to have a healthy development and aging, for example. So imagine that this person, this is the gene that is important, for example, for testosterone production in men. And you can imagine that even though that gene for testosterone is the same throughout that person's life, the actual amount of testosterone that is produced is going to be very different from that person when that person is a toddler or a teenager or in their middle or old age. And that's because, again, even though the sequence doesn't change, the tags, where they stick, how many of them there are, etc., will dictate to what extent that gene will be activated or not at different times in that person's life. So these are really essential processes just for your body to function. But what is, I think, has been one of an amazing sort of discovery is the fact that this is not just a very set biological process that is shut off from anything you do, but that actually your environment, what you experience in your life, may affect where and how many of these tags are on your DNA. And one of the first bits of evidence that came for this actually came from, again, identical twins. This is an interesting model, of course, because identical twins share, the same DNA sequence pretty much. So studies started looking at how similar are identical twins also in their epigenetic patterns. And what the studies found was that, yes, identical twins are more similar in epigenetics than, say, fraternal twins or siblings or two strangers in the street. But as you follow them in time, as they grow older, they actually become less and less similar in these epigenetic patterns. And this provided the first cues that actually that could be that could reflect the influence of these twins' unique environmental experiences, so that as twins grow older, they start to become exposed to more and more different things. They might go to different schools, make different friends. One might start smoking, the other one not. And all of these accumulated different experiences may contribute not only to these twins looking increasingly different and behaving increasingly different, but actually being different at the level of how their genes are functioning. And this has led to a huge interest in what kind of environmental factors actually might get under our skin and modulate the way our genes work. And here I'm just showing you a collection of different types of factors by now that have been shown to have associations. And I'd like you to take it with a pinch of salt, because this is a field that is exploding absolutely in this past decade. A lot of interest in what we call environmental epigenetics, so seeing what factors relate to epigenetic changes. Many of these studies are still based on sort of smallish sample sizes, maybe single studies. And a lot of these associations have not been replicated yet. So I just want you to look at it with a bit of caution, but there certainly are some factors that are emerging to be very robust and very strongly related to your epigenome. And some of these are also relevant for mental health, neurodevelopment, and also ADHD itself. So, for example, I think arguably the environmental factor that I think anyone in the field will recognize has the strongest or clearest impact on your epigenome is tobacco smoking. So tobacco smoking is associated with all sorts of changes to these chemical tags. And the relationship is so robust that actually algorithms have been developed that can predict based on someone's blood sample and these chemical tags in the blood sample, whether someone is a current smoker, a former smoker, or a never smoker, just based on epigenetic patterns alone. So the relationship is very robust. However, we also know now that diet, for example, is a really important factor. And this is because actually some of these chemical tags I mentioned, for example, these methyl molecules, they actually your body needs essential nutrients to be able to actually make these tags. So essential nutrients, things like folate, vitamin B12, are actually things that your body cannot produce naturally by itself. It needs diet to be able to get these essential nutrients. And these are what we call methyl donors. They actually are really important for donating, let's say, or for enabling your body to make these chemical tags that then go and stick to the DNA and change the way that your genes work. So there's quite a direct, clear link between dietary factors and your epigenetic regulation. But what I want to stress here is that it's not just these very physical exposures like you smoke or if you eat that have been associated with the epigenome, but actually also psychosocial factors that we know are really important for healthy child development and mental and physical health, such as the early caregiving environment, such as parental caregiving, but also on the other side, things such as early life stress or trauma exposure have been found to associate with epigenetic patterns. There is an emerging or growing literature in humans, and certainly it's something that's been shown quite clearly based on animal experimental models as well. One thing I want to stress here is that I'm talking about these factors as if it's a negative thing, negative environments having a bad effect on your epigenetic regulation. But I also want to mention that epigenetics is just really an adaptive response of your body to try to adapt to its environment. And while some of these responses might be helpful for the body to adjust to its current environment, some of them might actually be on the long term increase disease risk. So this is really an adaptive mechanism so that both negative but also positive environments might have an influence as well. So one question is, what type of environmental factors can affect the epigenome? The other question is, when in development or when in someone's life can this have an effect? And research so far shows that it's not just things that happen after birth, but actually the prenatal environment is very important. And the factors that occur in utero have been related to epigenetic patterns in babies as well. So I mentioned smoking, for example, like own smoking having an influence on epigenetic patterns, but actually you also see quite a strong signature in babies who are just born. So looking at epigenetic patterns in cord blood, you can see quite clearly if a baby has been exposed to smoking in utero in prenatal life. So prenatal factors also matter. Now, there is a growing literature in the experimental research. So this is something that is very controversial still, and we really don't know in humans to what extent this really applies or generalizes. But animal studies where, it's very tightly controlled and all of these animals are sort of genetically identical, and you can really, researchers look at the effect of one specific exposure, leaving everything constant. So it's not very representative of how things work in natural or real life. But what they found increasingly is that actually exposures might have an impact also preconceptionally. So factors like pollutant exposure, smoking, dietary factors, stress might have an epigenetic impact on a person's, an individual's germline. So that would be sperm or oocytes for women. And some of these signatures may be passed on after conception to offspring and potentially multiple generations. But as I said, this is still very controversial, and we just don't know to what extent this really applies to humans yet. So how is this actually relevant to health? I mentioned that epigenetics is sort of this cross-link between your genome and your environment, but why do we care in terms of better understanding development and health? Well, I think if you think of these epigenetic patterns as being so important for allowing your body to use its genetic material flexibly in different parts of your body and at different times of your life, you can imagine how actually alterations or changes in epigenetic patterns might lead to an increased risk of disease or poor health outcomes. And this is exactly what has been shown. So there's been a lot of studies now showing a link between epigenetic alterations and things such as coronary heart disease or diabetes or autoimmune disorders, but also increasingly brain-based and neurodevelopmental outcomes, including, as I will show you in a minute, ADHD. Although the application of epigenetics to mental health and brain-based outcomes is much more recent than in other areas of health. And in fact, I wanted to show you briefly, I want to highlight oncology. So this is really cancer research and clinical care, because this is one area where really epigenetics has made great strides in the past decade. So there's a very strong link between epigenetic dysregulation and various cancers. And I know this is not the topic of interest here, but I just want to show it because I think it's one area where really epigenetic applications in the clinic are starting to emerge. We don't know to what extent that will happen for other types of health outcomes, but it gives you an idea of what the future might hold. So with regards to, in the context of oncology, what you see is, for example, there's very recent evidence that shows that taking blood from a blood sample, you can actually detect epigenetic signatures from very, very early stage tumors, not blood tumors, but actually originating in different parts of your body, but that might release their DNA into the bloodstream, and that you can actually capture potentially a signal of these very early tumors months or even a couple of years before conventional early detection methods. So clearly that would have huge implications for screening and very early interventions, and ideally, increasing chances of survival and positive outcomes. Also in diagnostics, now epigenetics is starting to be really used clinically to enhance classification and diagnosis of brain tumors. So these are very difficult sometimes to diagnose accurately, because they can look quite similar but actually be biologically quite different. And at the moment, most of the diagnostics are based on histological assessments, which can be prone to error. So epigenetics is being used as a way to biologically informed way of classifying and separating different tumors that might actually need very different strategies. So for some of these, you might need to operate, for some not, or you might need to deliver certain interventions. So it really matters to diagnose these properly from the beginning to avoid making the wrong choices downstream. Also, what studies have shown is that people who respond versus non-respond well to certain cancer therapies show differences in their epigenetic profile. So ideally, this information could be used to actually try to tailor certain treatments in a more personalized ways to help choose therapies that will work best for specific individuals based on these characteristics. And finally, the first FDA-approved cancer drugs that are actually targeting the epigenome, so that is the target of these drugs, is to make epigenetic changes, are starting to come onto the market. But I think there's also a bigger potential. I think there's a lot of interest in the potential of epigenetics as a tool for broader health profiling in the population, not necessarily when individuals are coming to the clinic already with certain with a diagnosis or with a onset of a particular disorder, but beforehand, because the idea is that these epigenetic patterns, to varying extents, could be capturing genetic liabilities, a record of environmental exposures at different time points, your rate of biological aging, various health parameters, all of this, they could maybe give us an opportunity to better stratify individuals based on their potential risk profiles, and therefore deliver, better and more personalized risk prediction, health screening, and monitoring strategies. So now I come to maybe, I think, the part that might be most of interest to the attitude community here. And that's, well, how does epigenetics actually relate to ADHD? And I have to just be upfront that this is really a very, very, very new area of research. There have been some studies done in the past years, but the most powered, well-designed studies are only emerging now in the past couple of years. So there's certainly more questions than there are answers. So I only have a couple of slides, but I wanted to highlight really some of the interesting findings that are starting to emerge from this work. So most studies actually at the moment have been based on clinical samples. So here you have, for example, a group typically of children. I think there's been, 7 studies published so far with a very variable sample size. The smallest study was only 14 individuals. The largest was 500, but divided between a group who had already received a diagnosis of ADHD versus a control group. And usually what these studies do is that they take a sample, maybe a blood sample or saliva or buccal sample, and try to understand are there any epigenetic differences between these two groups, diagnosed with ADHD versus controlled. And here I want to pause 1 second to tell you this is important. So most studies, all studies in this area have to rely on easily accessible tissues, blood, saliva, buccal, tissues that we can access in living individuals. But these are not the most relevant tissues for what we're interested in, right? Because ADHD being very related to neurodevelopment, what you'd want to be able to do is see what the epigenetic patterns are in the brain, but that is just not possible clearly because we cannot just get a brain sample and look at epigenetic patterns. So we have to rely on peripheral samples. And this is important because we just don't know to what extent what we see in blood really reflects what's going on in the brain, which is the main organ of interest. So it might be useful for prediction or for, for various clinical applications. But if you really want to go to mechanisms, this is a challenge because we don't know what's happening in the brain itself. But basically what these studies do is usually they perform what we call an epigenome-wide study. So this is an example on the right-hand side. This is not an actual figure related to epigenetics and ADHD. It's just an example of what we call a Manhattan plot. This is usually what you generate from these studies. And basically you can see all these little dots. And each dot, you can think of it as one specific spot on your gene, on your DNA sequence, and it's asking, it's different spots where there can be these little the little epigenetic tags that I was talking about. So from your first chromosome all the way to your last chromosome. So it's like a map of associations for each of your chromosome and each of these dots. And the higher the bar, the more significantly associated that particular location in your genome is to ADHD, for example. But because we're making a huge number of comparisons for each individual, we measure 500,000 to 1 million different chemical tags across the genome. We have to be really, really strict for what we consider to be a significant association, because otherwise there's very high risk of false positives. So you see a little line here. This is actually the line that we say, okay, anything above this is what we consider a significant finding. So this is just to explain what an epigenome-wide study is. And so far, these studies really have not shown a very, very strong signal. So there certainly are associations between epigenetic marks and ADHD in these clinical samples, but they're not very strong. Most of these have not really replicated across these different studies, but there are some exceptions. So for example, there's this one gene called VPR2. This has the chemical tags on this gene have actually been identified by several of these studies, so it really adds confidence that it could be implicated in ADHD. And this is a gene that produces a small neuropeptide, so it acts as both a neurotransmitter as well as a hormone in the brain. And previous studies have shown that it is important for regulating mood, behavior, and circadian rhythm. And actually, studies have also shown that it is epigenetically, this gene is actually sensitive to a number of early environmental influences that we know are also implicated in ADHD. So the epigenetic status of this gene might be potentially also regulated by your environment. And interestingly, these studies have shown that actually there seems to be a stronger relationship between the epigenetic marks in this gene and ADHD in males compared to females. But at the moment, it's quite unclear whether that's reflecting a true biological sex difference, for example, or whether it reflects the fact that clinical samples tend to have many more males. So the group of individuals with a diagnosis of ADHD, they tend to usually have many more males than females, because that sort of reflects, particularly in childhood, the different likelihood of receiving a diagnosis with boys being more likely to receive it than girls. But one of the biggest limitations, I think, of these existing clinical samples is that you're really taking these epigenetic measurements only at one time point, and in individuals who have already been diagnosed with ADHD. So it makes it really difficult to establish what the relationship between these are. Is it that the changes in the epigenome lead to the development of ADHD, or at least precede the onset of ADHD? Is it that being diagnosed with ADHD might actually affect your epigenome mainly because of medication use or other lifestyle changes that are happening? Or what is happening? What is the timing and direction of these associations? And that's really, really important for us to disentangle, because depending on that answer, we will be better able to know how we might actually be able to use epigenetic information in the future in the clinic. So to address this complicated challenge, what our team in Rotterdam, as well as others around the world, are increasingly doing is to actually leverage the power of prospective birth cohorts. So these are not clinical studies. These are actually studies of the general population. So I can give you an example. I work in Rotterdam within one of these studies called Generation R. And we've been following the lives of 10,000 kids born in Rotterdam. Actually, we've been following them from the first trimester when they were in the womb in pregnancy, and now they're around 20 years of age. So we've been following them for 20 years. And along the way, because you're following the same individuals and their families over time, over these very important first 2 decades of life, you're collecting information along the way on a lot of relevant factors. So you collect information on genetics, on prenatal and postnatal environmental and social factors, and also collecting maybe blood or saliva samples, which enable us to measure these epigenetic factors, for example. So this really allows us, or starts to allow us, to disentangle the temporal relationship between your genes, your environment, your epigenetics, and ultimately your neurodevelopment and behavior and later health. The other advantage is that population studies don't focus just on individuals who have been diagnosed with ADHD, but allow you to look at the full spectrum of behaviors related to ADHD and core symptoms like inattention and hyperactivity as a spectrum, which we know it is, much like many other traits, whether that be depressive traits or blood pressure. These are really continuous traits. And at the very tail end, usually that's where diagnosis happens. But there's actually a big spread in the population. So we can look at the full range. And we can also look at ADHD in a more balanced way, in the sense of in the population, you have obviously a split, 50-50 split of males and females. So that allows also a little bit more of a balanced view on potential sex differences. So I know I'm short of time, but so I had to choose what to show. And I wanted to show you for me, I think has been the most surprising and thought-provoking finding in this very new research on epigenetics and ADHD coming from these large general population studies. So as I mentioned, one of the key advantages of these longitudinal studies is that you can actually measure epigenetics repeatedly over time, not just at one time point. And we can start doing that at birth. So before the onset of any ADHD-related behaviors, which allows you to start to separate a little bit more the timing of associations. And we can do it repeatedly so we can see if any of potential associations are actually stable or dynamic over time. And what we found, this is a finding that we first came across in one of these studies, because at the time, about 5 years ago, it was the only study in the world that actually had access to repeated epigenetic information, because it's very expensive and labor intensive to collect. But now we've replicated this finding in a much larger set, so in over 2,500 children from 10 different studies around the world. So we know that there is definitely something robust about this. And what we find is that actually epigenetic patterns at birth associate prospectively with the severity of ADHD symptoms in mid to late childhood. So you see a relationship of the chemical tags at birth with how severe ADHD symptoms will be 6 to 10 years later. But what's also really surprising is that actually you don't really see a strong relationship when you're measuring these epigenetic patterns at the same time as the ADHD related behavior and traits. And this is surprising because usually in longitudinal research, you expect that when you measure things close in time together, they tend to be more strongly related than if you measure them 5, 10 years apart. But this was really an exception to the rule. And I want to show you these Manhattan plots that I just explained. So these are real Manhattan plots from this research. And you can see here this epigenetic signal that we find at birth in predicting ADHD symptoms 10 years later compared to when you measure them at the same time point. And you can see that at birth, you have a stronger signal overall, not just in terms of things, locations in the genome that go above this very strict significance threshold, but the general strength of the signal overall seems to be stronger than in childhood. And when we look at birth and what these genes actually are, what is happening up here in these more strongly associated sites, what you see is that these are genes that tend to be involved in, play an important role in neurodevelopment and cognition very early on. And in these studies, one of the genes that has come up as I think one of the most interesting and really, I think, high confidence is ST3-GAL3. Now, this is a gene that actually completely independently has been identified as one of the strongest genetic hits for ADHD. So genetic studies, not epigenetic studies, but studies that look at actual DNA sequence variation have found that variants in this gene are amongst the most strongly associated with ADHD. And now we're showing from an epigenetic perspective that the chemical marks on this gene also associate with ADHD, particularly early onset and more chronic course of ADHD. We also know that these chemical tags on this gene are sensitive to some extent to environmental factors, although we need to map this more carefully. And in terms of function, this is a gene that encodes an enzyme that is very important for brain development, particularly myelination, which is basically like the sort of fat that insulates your brain cells to allow more speedy and efficient brain connections. And I think one of the things that adds confidence to this gene is that actually animal experimental studies have shown that if you knock out this gene, so if you turn it off experimentally, animals tend to then develop inattention and hyperactivity, which of course are two hallmarks of ADHD. So this is a bit of a flavor of some of these very initial findings that are coming in the area of epigenetics and ADHD. And I just wanted to reflect on this. And in the last part of this webinar, just single out, I think, 3 of the biggest challenges that we have at the moment clinically, or in terms of how ADHD is detected and managed and treated, and how in future, as this field grows, we might be able to use epigenetic tools to help address this. And of course, I'm speculating here. I don't know if this will really happen, but I wanted to single out things that I do think have a real realistic possibility of happening in the future. So one challenge is early detection, right? Because as I mentioned at the very beginning of the talk, risk factors for ADHD, they show very poor specificity. They're not specific to ADHD itself in most cases, and they show very poor predictive utility. So they're not very useful in the clinic or applicable at the moment. Early detection still relies at the moment on the presence of ADHD symptoms. So actually symptoms having already emerged. So that limits opportunities for very early or preventive interventions. So I think that potentially epigenetic markers could be used not as a tool in themselves to perform risk prediction, but more as part of a clinical toolkit that includes genetic information, environmental information, and also epigenetics to improve risk prediction models before symptom manifestation, opening potentially the door to new ways of detecting risk, particularly of impairment related to ADHD earlier and better. And in this respect, I think the finding that I just presented, where you see a signal of epigenetic signal at birth that seems to carry some level of information about the severity and development of later ADHD symptoms could be particularly interesting in this respect. And now we have a European project that's really focused. It's a 5-year project. It's entirely focused on understanding this and really seeing to what extent is there something here that could be potentially useful to leverage? So some of the questions we're trying to ask is, okay, how much of ADHD symptoms can be explained by these very, very early signatures? How specific are these signatures to ADHD compared to other brain-based or broader physical health outcomes? Our preliminary data so far suggests that there is a degree of specificity. For example, for things like BMI or obesity, you just don't see this relationship. If you measure epigenetic patterns at the same time as when you're measuring your BMI, there's a much stronger relationship than anything you can predict at birth. So there is definitely a degree of specificity, but we really need to map this out more comprehensively. We also want to know whether the signal could be informative for helping us. It seems a little bit stronger potentially for individuals who will end up developing an earlier onset and more chronic form of ADHD, but we need to look at this more carefully. Can we really utilize this to stratify a bit more individuals in terms of their likely course and chronicity of ADHD? And most importantly, why is it there in the first place? What kind of factors is it reflecting? Is it genetic liability? Is it prenatal environmental factors? Or is it actually representing the combination of these factors, which is what we've been missing, and what the promise, let's say, of epigenetics really is to capture both your genes and environment and their interactions. But one thing I want to say here is that even if we address all of these questions and we find that this is a useful tool, clearly there's very important ethical implications that we need to consider, because we're not talking about any diagnostic tool at birth. Far from it. I don't think that's really going to happen ever, in my opinion. But we're talking about risk prediction. And risk prediction, especially at birth, this is a very ethically gray area, because like any screening or risk prediction, you have risk of false positives. You have risk that any biological sort of risk might not actually ever materialize into any impairment. And so that could, there could be unnecessary measures or interventions that are administered. And there's also the question of actionability. What should we actually do if there is a positive screen, for example? And what's the acceptability? What's the acceptability for parents, for individuals, the ADHD community, for clinicians, public health, etc. So these are a lot of questions that we will need to address. First, we need to see if there's even anything there really that's actionable. But these are definitely questions at the forefront of my mind, at least. So the second, I'm almost finished. I think I'm probably starting to run out of time. But anyways, the second challenge, I think, is that of stratification and subtyping. So individuals with ADHD, currently, we know it's not a unified group of individuals. There's huge variability in onset, course, level of impairment, comorbidity profiles, and we just don't really know at the moment, even though we've mapped as a scientific community all of these risk factors, we're not good at the moment at separating and understanding why people follow such different courses and such different clinical presentations. So I mentioned in the area of oncology, using epigenetics to stratify. I don't know if this will be possible with ADHD, but it is maybe epigenetic patterns in future could help to identify more homogeneous subgroups or subtypes of ADHD, which might actually require quite different strategies and treatment options. And finally, that leads me to treatment. Now, we know that current treatments do work well for a lot of individuals, but a sizable minority of individuals, about 30%, don't respond so well. And so we really need better tools to help clinicians formulate more personalized treatment plans and better predict likely treatment response. Because at the moment, like in many areas, particularly in psychiatry and neurodevelopmental conditions, there's really this cycle of trial and error, trying first-line treatments, seeing if they work, otherwise trying second-line treatments, etc. And this is tremendously burdensome and stressful on individuals. There's high, the potential for side effects and for time wasted and all of these things and resources, all of these things, if we could just improve them. And this is where pharmacogenetics, I think, is what it's trying to do as a field, using genetic information, but potentially using supplementing genetics with epigenetic information could help to get at a better picture of an individual's profile and try to narrow down potential effective treatments accordingly. So it could be useful in future as a decision support tool to help predict and monitor therapeutic responses. And because epigenetic markers can point to potential causal mechanisms, they could also lead to entirely new types of interventions in the future. So with that, I'd like to conclude. I hope that you found this webinar interesting, and mainly that it's shown you how epigenetics does offer promising unifying framework for understanding how genes and environments actually come together at a biological level to shape children's development and health. There are certain areas of healthcare, such as oncology, that are seeing huge strides in actually the application of epigenetic knowledge and information to the clinic. The application of epigenetics in the context of ADHD is still extremely new, only in the past couple of years. So it's early days. We just don't know yet. We're still trying to characterize associations, seeing where it could be helpful. So we just don't know exactly how and whether it might be able to be applied. But already what it's showing us is it's improving our understanding of the likely biology by pinpointing particular genes that might also be responsive to the environment and implicated in ADHD. And also the really importance of timing, that these are very dynamic processes, and that in order, when you measure these epigenetic marks might make a huge difference in whether you're able to detect a signal or not, with, for example, at the moment, epigenetic patterns at birth seeming to be particularly informative, maybe because they're just capturing prenatal and genetic effects more cleanly at birth. And then as you go over time, there's a lot of other factors that happen and that signal washes out, for example. That could be a possibility. But ultimately, I think that what the hope is that epigenetics might be used one day as part of a future clinical toolkit for improved early detection, monitoring, and treatment formulation, bringing us one step closer to more precision medicine approaches. So with that, I'd like to really thank you. I've also put some references there that I found. Well, one is of a review we performed, but I found generally very interesting and helpful. And I think that they're all open access, I think, and they will be informative to the community if you want to read more about aspects of what I presented today. So thank you very much.
Thank you, Dr. Cecil. So before we start the Q&A, I'd like to share the final results from today's poll question, which was, what do you hope epigenetic research will one day lead to? And here's what you said. 24% said more personalized treatment plans. 23% said how the environment and genetic factors cause ADHD. 20% said how conditions can be prevented. 19% said improved screening and diagnostic tools, and 13% said the identification of more subtypes of ADHD. So now to your questions from our audience. We got some great questions. I've been hearing that genetic testing is a good tool to help determine which stimulant medication might be most helpful for an individual with ADHD. Recently, a clinical psychologist suggested we request this testing for my 22-year-old son who has inattentive ADHD. I'm curious to know, is there scientific evidence for this? And if so, could we request this testing from our GP, or do we need to see a specialist?
This is, a really important question. It taps into pharmacogenetics, and indeed it's an area that's fastly growing. My understanding, and there might be also some country-specific, differences in healthcare and what's warranted. My understanding is at the moment, pharmacogenetics in the context of ADHD, there is insufficient evidence in most cases to warrant it. From what I understand, mainly what it does is it can look at different genes that are just important for metabolizing generally substances or different types of medications, and they can tell you whether you might be a fast metabolizer or slow metabolizer, and that could have implications for dosing. So, for example, if you're an ultra-fast metabolizer, the standard doses that are given to a population might not work very well for you because biologically you have this higher rate of metabolism. So I think at the moment, the applications are more in terms of dosage, maybe some tweaks in the type of treatments, but from what I understand, there's not enough evidence base to roll it out, I'd say, at a population level. I don't know if the clinician has suggested it specifically in this particular case. There might be good reasons. I think it can help in some individual cases. So I would say, I don't know if this is covered in the US or in the country of the person asking the question by, standard healthcare, but to approach probably a clinical geneticist for this kind of a question.
Okay. Is there research on reversing the epigenetic pathway of ADHD? That is, if genes were altered by their interaction with a harmful environment in utero and in early life, can a quote unquote reverse direction pathway be found and developed to restore genes to a healthier state?
Really good question. Controversial, I think. So certainly there's no evidence at the moment specifically in regards to ADHD, because as I showed, just generally the field of epigenetics applied to ADHD is very new. So we're just trying to characterize associations in the first place. So we don't know to what extent what we're seeing is actually causal and whether it could be reversed. But more generally, there has been an interest in the potential of reversing environmentally induced epigenetic changes. Now, there are some studies showing that dietary factors, so like diet and exercise and sleep and improved mental health and physical well-being, and even therapy, there's very preliminary evidence that certain types of therapy might help to generally restore epigenetic patterns. So this is not through any chemical, actually, changing your epigenetics, but more through lifestyle interventions. And also in animals, we're collaborating with a group that's showing that in animals exposed to very severe early life stress, environmental enrichment, so this kind of giving appropriate care and space for these animals to play and more social interactions can help to reverse some of these epigenetic changes. Now, when it comes to actually targeting the marks themselves chemically, we're not there yet. I mentioned with cancer treatments, there are some initial drugs that are starting to target epigenetics, but it's really not well researched yet. And there's a high potential for off-target effects because we don't really know what is normal epigenetic, what are normal marks at a given point in development or for a given individual. Really manipulating that could have consequences. And so we're not there yet, but more in terms of lifestyle interventions, there seems to be some support that could really help.
Okay. How can we learn about the epigenetic background of an adopted child where we have no information about birth parents?
Well, that's interesting because for the epigenetic patterns, as we said already, there's certain things that we can measure with some degree of confidence. I don't mean really at an individual level, but at a population level. If you take epigenetic samples from a group of people, you can sort of estimate some of the exposures that they might have had from pregnancy on, as early as pregnancy. But how that relates to individuals being, adopted, I think there's no certainty still yet to really have that picture of what epigenetic patterns are really reflecting your genes, which ones are really reflecting your environment. So I don't think we're at the stage yet where it could help to get a record of exposure of a specific individual at this time.
Okay. We had some questions around generational or intergenerational trauma. Can intergenerational trauma contribute to ADHD? And where there is trauma or other environmental exposure to trauma, how many generations might this last?
So I touched on this really briefly in one of the slides, but there is a lot of interest, again, as to these potential preconceptional effects. Now, the current opinion holds that in most cases, at conception, a lot of these epigenetic patterns that you may inherit from your mom and your dad get reset. So there's multiple waves of reprogramming that are happening during fetal development that are really important just for fetal growth and, in a sense, are giving a baby a sort of a clean slate for their own life and their own epigenetic regulation. However, there is evidence and growing evidence from animal models that some of these let's say legacy epigenetic marks might actually escape this reprogramming and could potentially affect development and health in several generations. So some of the studies, as I mentioned, they're very tightly controlled. So, it's very difficult to make a parallel in humans because in humans, we are genetically different from one another. There's a million different environmental exposures that might interact with one another. In animal studies, it's really everything is held constant and just that one exposure, whether it's air pollution or stress, is the one that gets that's the one of interest. But these marks can actually be passed on for around 5 generations in some of these studies. But it's very difficult to establish whether that's the case in humans at the moment.
Wow, 5 generations. Wow. Someone asked, can you speak about comorbid conditions such as autism or anxiety disorders in relation to epigenetics of people with ADHD?
So not currently of comorbid conditions within specifically individuals with ADHD, but generally there's a lot of development now. Obviously, part of epigenetic research is focusing specifically on ADHD, but also there's more and more studies that are characterizing associations between epigenetics and a vast number of other conditions that tend to be often comorbid with ADHD, like anxiety or depression, or neurodevelopmental conditions such as autism. So there's a lot of associations that have been reported. Again, we're still at a stage of trying to do these large collaborative studies with many different partners around the world to try to identify the most robust and replicable associations. So there is definitely evidence, for example, for schizophrenia and emerging evidence for autism that there are potentially also these very early epigenetic signatures from birth. And that is consistent with the idea that some of these processes may be happening in fetal neurodevelopment, and that's why they might be seen that early on.
Someone asks, are you aware of any research that examines the role of diet in treating trauma, especially inherited family trauma, as a result of epigenetics? Could changing diet help with overcoming inherited family trauma?
I think there's generally an interest in diet and its effect on epigenetics in these experimental models. That was part of that kind of environmental enrichment I was talking about that did come from early and maybe even preconceptional exposure to trauma. And there is some supporting evidence there. But in humans, again, it's difficult to measure because you would need to have information on people's exposures and their epigenetics in multiple generations within the same study. And that's something that now it's starting to be possible. Like some of the longest running studies now are beginning to have epigenetics in 2 generations and even 3 generations. So in the near future, I think we'll be able to address that more, but we really need multi-generational epigenetic data to really look at that closely.
Okay. Is there any research that you know of on fertility drugs and ADHD?
Fertility drugs and ADHD? No, maybe that's also not, I might not be aware personally because it's not my area of expertise. I do know that there is interest in fertility aid and epigenetics, but I don't know specifically, at least in the reviews that I'm familiar with, that hasn't been, I don't know, maybe because there's not enough research or because there's not enough supporting evidence, but it's not one of the most prominent risk factors that are put out there. Yeah.
Okay. What if the mother quit smoking years before pregnancy, but the father smoked during conception, but not around the mother during pregnancy? Would that contribute to a risk of ADHD in the child?
Very tricky to answer at the moment. Yeah, I think very tricky. These are very, very important questions, but it requires data where you would need to know exactly a person's record of smoking exposure and if it happened before or after conception and how that might affect the next generation. Again, these are things only at the moment animal models can do. And there has been evidence that smoking influences epigenetic patterns in sperm. But how that relates to not being exposed to smoke during pregnancy, but still having those preconceptional effects in humans is very difficult to address. So hopefully watch this space, and over the next decade, I think, or maybe in the next few years, there'll be more insights as these multi-generational studies become more available.
This next question might fit into that. What can an adult male with ADHD do to reduce his offspring's ADHD risk? Well,
that would require a lot of knowledge on causal factors. And I I think that's a bit disappointing in a sense. But as I mentioned, even though a lot of risk factors have been identified, which ones are truly causal is still a bit of an open question. I think just general health advice, like not smoking and eating healthily and stress management, exercise, but these are quite general strategies, but that are important for multiple aspects of health, mental and physical health. But in terms of very specific factors to ADHD, at the moment, we don't really know causal factors that are so specific to ADHD to provide that personalized advice.
Well, unfortunately, that has to be our last question because we're out of time. But Dr. Cecil, thank you so much for joining us today and for sharing your expertise with this ADHD community. Very much appreciated.
Thank you so much for having me.
And thank you to today's listeners. We hope to see you again next week. Make sure you don't miss future Attitude webinars, articles, or research updates by signing up to receive our free email newsletters at attitudemag.com/newsletters. If you're listening in replay or podcast mode, visit attitudemag.com and search podcast 473 to access the webinar resources, or simply click on the episode description wherever you stream your podcasts. And if you support the work we're doing here at Attitude to strengthen the ADHD community, we encourage you to visit attitudemag.com/subscribe and sign up for Attitude Magazine. Sign up today for Attitude Magazine for yourself or to share with a teacher or a loved one who could benefit from greater ADHD understanding. Thank you, everyone.
For more Attitude podcasts and information on living well with attention deficit, visit attitudemag.com. That's ADDITUDEMAG.com.
O'Reilly. You've got questions? O'Reilly Auto Parts has answers. Need a pro you can trust? We've got that too. No matter what you need, our professional parts people have the training and expertise to help you do things right. Deep automotive knowledge. Just one part that makes O'Reilly stand apart. The professional parts people.
O'Reilly.